What I'm starting out with here is going to be our double-stranded DNA molecule, all right? In terms of the double-stranded DNA molecule, there are a couple things I want to point out as far as how that all comes together before I show you how the linkages happen, okay? But in terms of that, remember I said this was our 5' carbon over here. So in terms of that, where you've got the free phosphate, meaning it's not making a covalent bond with another structure, that's going to be the 5' end of our DNA molecule. Where we've got a hydroxyl group, all right? Remember, this was our 3' carbon. So this is our 3' end, all right? DNA is going to orient itself in a 5' to 3' direction, all right? So you can see I've got 2 nucleotides here. I've got 2 more nucleotides over here. We've got a free phosphate. Free phosphate means that's the 5' end. Hydroxyl group, this is going to be our 3' end, all right? So if you notice, let's move that 3 over just because I'm going to be anal about it. 3', all right? We're going to go 5' to 3'. So when we talk about DNA and you hear 5' to 3', that's what it's referring to, all right? And if you notice, the 2 strands that I drew here go in opposite directions, all right? That DNA is going to be anti-parallel. That's what that means, okay? And in terms of that now, we're going to have within that double-stranded DNA double helix, all right, we're going to have the connection between our nucleotide here and our nucleotide down here. I've already drawn this one connected, okay? But what's going to happen with our nucleotide here is we are going to form what's known as a phosphodiester bond, all right? You've got an ester group in here, that's why it's a phosphodiester, phosphate, ester, all right? Our phosphodiester bond is over here on this side of the molecule. That phosphodiester bond, again, is a type of covalent bond. So this is a really strong bond. You're going to hold those sugars and those phosphates together using a good bit of force. It's going to take a lot to break that apart, all right? And that's going to be important. The other type of bond that's going to be important with your DNA double helix now is going to be a hydrogen bond. And we're going to have hydrogen bonding that's going to occur in the middle, all right? And I'm going to draw the hydrogen bonds here as dots. And I'm going to draw them as dots because hydrogen bonds are not a very strong type of bond, so it doesn't take quite as much energy to pull them apart. That's going to become important when we talk about DNA replication, because in order to make a copy of the DNA, we're going to need something to start with. So with DNA replication, those 2 strands of DNA are pulled apart and are used as a template to make a new strand of DNA. So as part of that, you've got hydrogen bonds kind of holding the center of that DNA double helix together. And if you also notice, I've got some pairings here, all right? Adenine is always going to pair with thymine. Cytosine is always going to pair with guanine. If you also notice, I drew 2 dotted lines between adenine and thymine, 3 dotted lines between cytosine and guanine. That wasn't a mistake. That tells you the number of hydrogen bonds that are going to hold those 2 molecules together. So between cytosine and guanine, we've got 3 hydrogen bonds. Adenine and thymine, we've got 2, all right? the pairing of adenine and thymine, cytosine and guanine, is going to be Chargaff's rule. All right? So in terms of that, adenine will always pair with thymine, cytosine will always pair with guanine, normally with the DNA double helix. All right? You can get some odd stuff that happens with mutations. We're not going to worry about that right now. What I want you to remember, A is always supposed to pair with T, C is always supposed to pair with G. All right? And in terms of the chemical structure of DNA, all right, where this is going to become important, where we're going to revisit this, is when we talk about DNA replication. Because how those nucleotides are put together, how those strands are put together, are going to become important for when we talk about how that bacterial cell makes an exact copy of its DNA. All right? So thank you for your attention. Let me know if you have any questions.